Thank you so much for giving me opportunity to present our ongoing work on spatial regulatory landscape of glioblastoma tumor immune microenvironment. I want to start my talk with our biological modification. Glioblastoma is a very aggressive malignant brain tumor, and overall survival is only 8 months. And most of the patients, like less than 10% of patients, survive like beyond 5 years. And currently treatment options are like surgery, radiation and chemotherapy. So we still need better treatments for glioblastoma patients. One of the caveats related to treatment of One of the caveats of treating glioblastoma patients are like tumor heterogeneity, high cell infiltration behavior, and the blood-brain barrier. So in this study, our motivation is maybe if we better understand the regulatory programs of the immune tumor microenvironment, this can open new therapeutic avenues for patients. As we listened in the previous talk, like spatial omics is a very powerful technology. You can get molecular readout in a spatial context, and which can help us better understand the immune tumor microenvironment, glioblastoma, as well as other cancers. In this study, motivated by this, we mined publicly available glioblastoma samples and try to understand the immune tumor microenvironment. And in this figure, I'm showing you our biological motivation. As you can see, like signaling pathways converge on the nucleus, modulate the activity of transcription factors, which cause up and down regulation of target gene expression. And these gene expression programs define the phenotype of a cell. As you can see from this cartoon example, cells doesn't act in isolation. They receive signals from surrounding through the ligand receptor-mediated cell-cell communication or secreted molecules such as cytokines. So now with the spatial transcriptomics, we have a like molecular readout of downstream of these regulatory programs, gene expression, but this is high-dimensional data. Capture efficiency is low, so the data is very sparse. So we use our recent computational frameworks to summarize these high-dimensional data in terms of transcription factor and pathway activities. Let me show you how we are going to do this more formally. Sorry, the figure is a bit fuzzy, but let me first start with introducing the data types, data sources that we have. From the spatial transcriptomics, we have spot-level gene expression data, which is genome byte. But as I mentioned, this is like sparse data, so the capture efficiency is low. We can also get from the spatial transcriptomics like XY coordinates of where the cells are located. And most of the spatial transcriptomic technologies also has H&E stained images, which is giving us a morphological information. So we are going to integrate these two data sources with prior information, like transcription factor target priors or pathway priors. So this Y matrix is showing you the gene by spot level gene expression data. This is not, for most of the spatial transcriptomic technology, is not a single cell resolution. It can include like 1 to 10 cells from same or different cell types. And we also have these XY coordinates of these spots, as well as corresponding H&E stained images, which can give us a morphological information. And we use these data from this kernel matrix, which is giving us the similarity between distance, as well as morphological similarity between spots. And we also have this prior information transcription factor gene matrix or pathway gene matrix. So we are going to integrate these three data sources in a supervised approach. We are trying to predict the gene expression Y from this prior matrix, transcription factor pathway prior matrix. In doing so, we are learning this W, which are like inferred TF and pathway activities. So when we are solving this, we also impose these spatial constraints from this kernel matrix, as well as other regularizers, L2 regularizers. So this allows us both modeling the spatial and non-spatial dependent pathway and transcription factor activities. Now we summarize this high dimensional gene expression data in terms of transcription factor and pathway activities. Then we can use cell type deconvolution approaches or annotations from a pathologist to identify pathways and transcription factors specific to cell types, or like these spatial niches called meta programs, which I'm going to talk in the next slide. After we do this, also we can link these to like ligands and receptors to identify like therapeutic targets. Let me show you how we are going to do this. So as I mentioned, we mine publicly available glioblastoma data from there is 3 different sources. So there are total 27 samples. We also use priors, transcription factor target priors from HTF target database, as well as pathway priors from molecular signature hallmarks of cancers. And one of the previous glioblastoma study identified 14 different malignant and non-malignant meta programs. We are also going to use these annotations and try to find what are the transcription factors as well as pathways associated with these meta programs. Let me give you one intuitive example. Glioblastoma is, as I mentioned, very aggressive brain tumor that become hypoxic due to the rapid growth and abnormal blood vessels. And I'm here showing you, our analysis with just one sample. And in this plot, I'm showing you this annotation of these 14 different meta programs. And this orange part represents the hypoxic mesenchymal meta program. We first look, we know that HIF1 alpha is a main transcription factor driving the hypoxic pathways. First, we look at the inferred HIF1 alpha activity, and we are seeing that it is high activity in the hypoxic mesenchymal regions, and you can see in this widening plot is clear its highest activity in this region. Again, inferred hypoxia activity was also high in this region, but if you just look at the mRNA level, you don't see this distinction. There are multiple, as you know, transcription factor activities generally not correlate with their mRNA level. There is this post-transcriptional modifications. And from this example, you can see that our approach by integrating priors gene expression in a also utilizing the spatial regularizer, we can quantify the activities of transcription factors and pathways that are normally obscured by their modus mRNA levels based on spatial transcriptomics. Then we can perform a statistical analysis and get this association for each meta program within a sample as well as across samples. Here I am showing you with just one sample, and you can see that with this figure we can identify what are the key regulators for this meta program. These are like spatial niches. Here, for example, this mesenchymal hypoxic area, you are seeing high activity of HIF1 alpha, EPAS1, which is HIF1 alpha beta. But for example, in mesenchymal astrocyte, you are also seeing common transcription factors, but you are seeing these different ones like TCF4, which involved in Wnt signaling, which has importance in astrocyte development. Similarly, bottom dot plot is showing the pathways associated with these different meta programs. So we apply our method to all the samples in the data set, and we identified transcription factors, pathways associated in majority of samples with these different meta programs. When you look at this table, you are seeing, for example, SOX2, which has key roles, known key roles in glioblastoma like stemness, and we are seeing that SOX2 activity in high in mesenchymal astrocyte, hypoxic region, and mesenchymal-like malignant cells. Like also you can see that like hypoxic activity is just high in hypoxia area, but not in the other metaprograms. Or if you look at this neuron metaprograms, we are seeing high activity of HOX signaling, which has a role in like neural development. Next, we look at like whether we can identify, relationship between ligands, receptors, transcription factors, and pathways. And here I'm showing you the analysis with the one sample. In this heat map, I'm showing you the correlation between transcription factor, pathway, activities with ligand and receptor mRNA expression in nearby spots. And when we look at, we see like reasonable things. For example, SOX2 and HIF1 alpha transcription factor activities highly correlated with WIM and VEGF, as well as in terms of receptor CD44, RAK1, and all of these involved in the stemness pathway. And you can see this more clearly in the spatial plot. You can see that inferred SOX2, HIF1 alpha activity is spatially correlated with WIM and VEGF and CD4 and RAK1 mRNA levels. Then, we ask whether we are seeing these associations across the samples. Like SOX2 has been extensively incubated in glioblastoma malignancy, and we look at this correlation across samples, SOX2 activity with the other receptor ligands and pathways. Here you can see that from this correlation heat map, SOX2 is highly correlated with WIM mRNA levels across all samples, as well as CD44. And our collaborator look at the protein level in the GBM tissue samples. And here I'm showing you the immunofluorescence, like checking the protein levels with the immunofluorescence. Like here, we are showing just two samples as a proof of principle, like SOX2 protein expression, CD44 protein expression, and WIM protein expression. And you can see from the last image, they indeed co-localize in the immune tumor microenvironment. Similarly, HIF1 alpha, which correlates with increased tumor grade and reduced overall survival. We look at the across sample, we are seeing whether correlation with identified ligand and receptors. And we also saw HIF1 alpha highly correlated with RAK1 and VEGF. And indeed, in the protein level, we were able to show this. And you can see, actually, this is very heterogeneous. Actually, we saw that the correlations, we were able to detect the correlations because HIF1 alpha is associated with aggressive tumors, and we are seeing higher correlations in the aggressive tumors. We are very interested in also resource development, and we start developing this resource for the community to showing that spatial gene expression, spatially inferred TF activity, as well as pathway activity for one samples as well as across the samples. This is still ongoing work. If you can check our web resource, that we welcome any feedback from the community to make it better. With that, I just want to summarize. Today, I show you how we can integrate prior information with spatial transcriptomic data to capture robust biological signatures in terms of pathway and transcription factor activities. And I show you how, as a proof of principle in the glioblastoma. But this framework can be applicable to the various disease and biological systems. Currently, we are applying this from our collaborators and stage liver disease, as well as pancreatic cancer. For our method paper is under currently review, which focus on the transcription factor activities. If you are interested in, you can check our bioarchive as well as GitHub for the inference of TF transcription factor activities. With that, I just want to thank Linan Zhang and Matthew Liu. Matthew is an undergraduate student who created this web resource. And Lina is my former postdoc who I continue to collaborate with, who lead these two projects. I want to also thank all the other lab members, our collaborators to perform validation experiments, as well as our funding. Thank you so much for listening. I'm happy to take any questions.
Nice talk. I have a question. So you linked the transcription factors and ligand receptor interactions in the same spot, but I wonder, are there any evidence showing there are functional connections between transcription factor and ligand receptor interactions?
Actually, that is ongoing work with our collaborator. As a next step, we want to, make functional validation beyond the protein level expression.